Hi everyone. In this video, I'm going to show you how to design PCR primers, but not just any primers. In this specific example, I'm going to show you how to design some primers that will copy a sequence out of the genome, let's say a putative promoter, and then we're going to clone that sequence into an expression plasmid so that that promoter can, let's say, drive a gene for something like GFP. So let's start by talking about the cloning process itself. Now, for more information on this, you can go to our other video where I go into detail about PCR restriction enzyme cloning, but here's the overview of the process. First of all, you're going to start with a plasmid that has a promoter already, something like this, the EF1 alpha promoter, and we need to cut that out. So we can use restriction enzymes to do that. Let's say I'm going to use SAL1 and KPN1 here. They're going to bind to the DNA and cut it. And that's going to give me this binary mixture of these two fragments over here where I've cut out the old promoter, that's the EF1 alpha, and then I have the rest of the plasmid backbone here. And I can separate those two fragments on a gel. So the promoter is going to be smaller than the backbone. I can separate those on the gel here and cut out my backbone. And what that will give me is this plasmid sequence here, but wherever the restriction enzymes cut it, they leave these single-stranded overhangs that we call sticky ends. So those are going to be very important later, but for now, let's just leave it at that. And then the next thing I'm going to do is I'm going to copy my new sequence from some sort of template. This could be another plasmid. In this case, let's say it's a chromosome from the genome. So let's say we're amplifying a promoter from the chromosome. There I can use PCR, polymerase chain reaction, to copy billions, trillions of copies of that promoter sequence from the chromosomes. And at the same time, if I design primers correctly for this reaction, I can also modify the ends of that sequence as well. So if I use SAL1 and KPN1 to cut my destination plasmid up here, I can also add SAL1 and KPN1 sites to this promoter sequence. And then what that would allow me to do is, after I purify that amplicon on a gel, I can add those restriction enzymes to this promoter sequence, and it will give me the same sticky ends here on the promoter that I have on my plasmid backbone. So let's say there's a bunch of A's here, there would be a bunch of T's here. These two sites would be complementary, and they would hydrogen bond to each other if I mix them in solution. So I do just that. I mix my digested PCR product and my digested plasmid backbone. They will naturally come together in solution. Those hydrogen bonds will form, but they are transient. So I also add T4DNA ligase, and what that does is it covalently bonds these strands to each other. So these strands to those strands, for example, to give me a nice covalent linkage that's permanent. And that will give me my final plasmid right here. And that's all facilitated by these sticky ends here. Alright, but then honestly, that's a low yield process. You only get a few of these plasmids in each reaction. So you've got to take that and you've got to transform it into E. coli. So even if you have one of these plasmids, if one of these plasmids makes it into one E. coli cell, that E. coli cell will begin to divide. It'll make more of the plasmid, more of these cells, and we'll get something like a colony shown down here on the LB agar plate. And then we can take that colony, put it into some liquid media, grow up a lot of cells, and then the more cells I have, the more plasmid I have. I can take the plasmid back out, get a large amount of plasmid, send that off for sequencing, and then I can confirm that I do have the promoter in place. So it's the new promoter, not the old promoter, and there's no mutations in it either. That's why we do sequencing. Okay, so that's the basic overview. But you can see a very important step in all of this is being able to copy the promoter sequence out of the genome and simultaneously add these restriction sites to each end. So that's what we're going to talk about in this video, is how you design primers that will give you this product right here. But again, before we get into the primers, we have to explain how PCR works first. So in the polymerase chain reaction, you're going to start with some sort of template. Let's say it's an entire chromosome, something like that. And you want to copy just a single or an isolated sequence out of that chromosome. What you're going to do is you're first going to heat the solution to a very high temperature, something like 95, 98 degrees Celsius, almost boiling. And what that's going to do is it's going to melt apart the double-strand DNA here into single strands. And so once you have those single strands, what you can do is slowly cool the solution to this range right here, around 50 to 70 degrees Celsius. Now at that temperature, that is still too hot for these large strands to re-anneal to one another. They can't come together just yet. But smaller pieces of DNA, let's say somewhere between 20 to 50 bases, can attach if they are complementary at that temperature. And so this is where we add what we call primers, or short oligonucleotides of DNA. All right? So the key here is that these primers are relatively short. They're only about 20 to 30 bases, so they can anneal at this elevated temperature. And on the 3' end here, they are perfectly complementary to the target sequence. So that's how we define which stretch of the template we want to copy. We're going to have one forward primer here that binds to, let's say, the beginning of our target sequence, and another reverse primer that binds to the end of that sequence. So this forward primer is going to be perfectly complementary to the beginning of the sequence. The reverse primer is perfectly complementary to the end of the sequence. But one interesting thing about PCR is that these primers only have to be complementary on their 3' ends. On the 5' end, you can put almost anything you want. It doesn't have to be complementary to the target. So remember, in restriction enzyme cloning, I need to have restriction sites on my PCR product. So I can do that right here. I can add restriction sites for different enzymes, let's say SAL1 and KPN1, to these primers, and they will be conserved as this sequence is copied. Okay, so I've got a primer here. It's got two parts. The 3' end is complementary to the target. The 5' end is not. I can add restriction sites there. So anyways, so far I've melted the DNA, the template DNA, apart. I've cooled the solution down to 50 to 70 degrees Celsius to allow these primers to anneal. Then I slightly increase the temperature to 72 degrees Celsius. Now what's going to happen there is DNA polymerase will bind to this primer template duplex that we have, and then at 72 degrees Celsius it will start extending that primer. It'll start copying the template sequence onto the primer. So we get this primer extension here in the 3' direction, and this is going to happen for both primers, such that at the end of this cycle, my primers will have grown substantially to copy whatever was in the target sequence here. Okay? So if in this example I started with one copy of the target sequence, now at the end of this these steps, I have two copies of the target sequence, one in blue, one in green. And if I keep doing this, let's say about 35 times, I can get billions, trillions, even larger numbers of my target sequence here. And notice what's happening is that the new target sequence that I'm making here in green is always flanked by these restriction sites. So there you go. That's how PCR works. We melt the DNA apart, we have our primers that anneal, they define the sequence that we're amplifying, the beginning and end of it, that is. Then DNA polymerase comes in and extends the primers. And we do this over and over and over again, and we end up with our PCR product that is the target sequence plus whatever we want on the ends. Okay, so now let's actually look at how I would design those primers specifically. Let's zoom in to the base pair level. So here again, let's say this is a chromosome, and this target region right here is the promoter sequence. Let's take apart those strands and look at what the forward and reverse primer would actually look like. So I got my two primers here. First of all, the 3' end has to be complementary to the target strand. So the forward primer is going to bind to the non-coding strand down here. So its sequence is going to be, let's say, ATGT. You can see the A binds to the T, T binds to the A, etc. One very easy way to get that sequence is you can just copy whatever on the coding strand up here. So you can just copy ATGT. The reverse primer, though, it binds to the end of the target sequence, and it is the reverse complement of the coding strand. So if the coding strand is GATT, then that reverse primer needs to start AATC. So you got to flip things a little bit. You can't just copy this sequence from the coding sequence. Okay? So there we go. We're going to have our complementary regions there. They're going to be about 20 to 30 bases long. Even though I only have 4 bases shown there, they're going to be a little bit longer. All right, and once I have that figured out, I can add my restriction sites there. Now, these are just arbitrary restriction sites, but I do want to point out that they are not strictly complementary to the target sequence here. Remember, on the 5' end of this primer, I don't need to have complementarity. As long as I've got a good region of complementarity on the 3' end, I can put whatever I want on the 5' end, including these restriction sites. And then a curious thing about restriction enzymes is that they will bind to the sequence, but they actually need a broader piece of DNA to bind to so that they can get a good enough grip to stay on the DNA long enough to cut them. So it would be insufficient if I ended my primer here with the restriction sites. Instead, what I have to do is I also have to add what we call protecting bases. Now these would be 3 to 4 random bases that I put at the 5' end of my primer. And what that does is it just gives the restriction enzyme, when it comes in here to bind this site, more DNA to attach to. Some restriction enzymes absolutely have to have these protecting bases. If you don't add them, then they don't bind or cut at all. So it's always a good idea to just get in the habit of adding them, even if you have a restriction enzyme that doesn't absolutely require them. So these protecting bases can be anything, but I would like to point out that they should not contribute to the formation of any secondary structures. So for example, this forward primer here, one worst case scenario is that it could bind to itself instead of the template. So you would want to make sure that these ends right here didn't contribute to the formation of any, let's say, self-dimers. So this, for example, you can see here in the restriction site, this is a palindrome, TGTACA. So if I took this primer, flipped it over, the T would bind to the A, the G would bind to the C, and so on and so forth. So we could actually have some binding that happens here. So I wouldn't want to make that worse by making this N right here a T that would bind to that A, or an A that would bind to that T. So you kind of get what I mean there. I could just make these all C's and there would be no interactions, or sorry, no, I could make these all G's and there would be no interactions with these bases down here. Alright, so anyways, there you go. That's how you design these primers. Start off by making a 3' end that's complementary, add your restriction site and protecting bases to the 5' end. Oh, one more thing. On the 3' end and that region of complementarity, you should also, if you can, end that 3' end with 1 to 3 C's or G's. So remember, C's or G's form 3 hydrogen bonds with their base pairing partner. So that's nice to have in the PCR reaction because, remember, fusion polymerase or DNA polymerase is going to bind to this duplex and then extend in the 3' direction. If that 3' end is just, let's say, flapping in the wind and it's not solidly bound to the template, then it's going to slow down the polymerase. So I would much rather have C's or G's that have 3 hydrogen bonds. They're going to be tightly clamped on to the target sequence, more so than, let's say, an A or a T that only makes 2 hydrogen bonds. We're talking about elevated temperatures here, so those hydrogen bonds could break or reform pretty easily. So there you go. It's not absolutely necessary, but if you can choose a complementary sequence that ends in a C or a G, then that would definitely be recommended. Okay, so now let's jump right into the process. In this case, we're going to use this example from a previous video here where we have the transcript sequence for interferon lambda 1 that's shown down here at the bottom, and then the 1500 bases upstream of the start codon, the ATG. That's what we're calling the putative IFN lambda 1 promoter. Alright, so we want to copy this sequence out of the genome and clone it into a plasmid to see if it actually is a promoter, if it can drive expression of, let's say, GFP. Alright, so we need to copy this sequence right here. The first thing I'm going to do is I'm going to define the ends of that. So let's say I want to put my forward primer right here at the beginning of the sequence. However, I can tell by just looking at this sequence, you can see there's a bunch of T's here. That would actually create problems for me later on. So I'm not going to put my primer right here. Instead, I'm going to shift it in a little ways. Okay, so I'm going to lose a little bit of sequence here, but I'm going to lose also that poly T region there. Okay, so I'm shifting my primer down, and I'm going to start here, let's say. And that's something you might have to do. You might have to shift your primer downstream, upstream, to get the best primer possible. That's definitely okay, as long as you're amplifying most of the region here. Alright, so let's say that's going to be my forward primer. I'm going to start there with that GTAG. I'm just going to copy 30 bases there. That's a good starting point. Most primers are 30 bases or less, so we're going to copy those 30 to define the beginning of the sequence. And then for the reverse primer, I'm going to copy the 30 bases that are immediately upstream of the start codon. Okay, but remember, the reverse primer actually needs to be the reverse complement of the coding strand. So you can see what I have here, TGCCG. TGCCG is just verbatim what is in the coding strand. I need to take the reverse complement of that. So a very easy way to do that is to use a web tool, something like reversecomplement.com here. All right, and I can copy, let's see here, this sequence, paste it into here, hit reverse complement, and that's what I need. All right, so let's jump back into the PowerPoint presentation. There you go. So this is just the reverse complement of this sequence. You can see this one ends in CC, but this one begins in GG. That's what we mean by reverse complement. Okay, so now I've got two primers. But honestly, being 30 bases long, that's probably longer than they need to be. So the next thing I'm going to do is I'm going to truncate them. I'm going to shorten these primers until they both have what we call a melting temperature of around 65 degrees Celsius. So just to recap, if I go back to our slide on PCR, remember the first step is to melt the DNA apart, but then I'm going to anneal my primers. So right here, when I say melting temperature or annealing temperature, that's the temperature at which my primers start to bind to this template. Okay? So I want a melting temperature that's somewhere in this range of 50 to 70 degrees Celsius. Definitely no lower than 50 degrees Celsius. That can create a lot of problems for a PCR reaction that I won't go into right now. But I actually like to err on the side of caution and be higher in the melting temperature. I prefer to be around 65 degrees Celsius. So I'm going to say I want 65 C to be the temperature at which my primers bind. So that means I'm probably going to have a little bit longer of a primer than some other folks. But in my experience, that's what's worked best. Okay, so coming back into here, next thing I'm going to do is truncate those primers. So I can go to any number of web tools to estimate the melting temperature of a primer. In this case, we're going to go to New England Biolab's TM calculator. That's because for our PCR reaction, we're going to use their fusion DNA polymerase. So it's always best to go to the manufacturer's website. They know their enzymes. So we're going to select fusion polymerase here. We're going to use their HF buffer, and we're going to use a primer concentration of 500 nanomolar. Okay, so now that I've got those settings put into the system, I can take my primers, there we go, that one right there, and this reverse complement over here for the reverse primer, and this is what we see. So you can tell already the program is telling us that the melting temperature of these primers is higher than it needs to be. So I want to get down to around 65 degrees Celsius, so I'm going to start shortening these primers. The way I'm going to do that is I'm going to remove bases from the 3' end on the right side here. Okay, I can do that because the polymerase binds to this primer and then extends in the 3' direction. So whatever I remove here, the polymerase is going to replace. That's not true of the 5' end. I wouldn't want to take bases off of there if I could avoid it. Okay, so I'm going to remove bases from the right side here until I get down to, there we go, perfect, 65 degrees Celsius. So that would be my truncated forward primer. Now for the reverse primer, I do the same thing. Since this is the reverse complement, this is the 3' end. So I start to remove bases there. All right, and now I've gotten down to about 66 degrees Celsius. But I would like to point out that here, I do have a bunch of C's on the end of this primer, but it's actually too many C's. If you can get 4 or more C's or G's in a row, they can start to form some inhibitory secondary structures. So while it's good to have all that hydrogen bonding on the 3' end of my primer, it's actually too much of a good thing. You want to generally keep that to only 3 C's. So I'm going to take 1 more C off. That gets me down to 3 C's. It does take me a little bit below my ideal melting temperature, so I'm down to 64 now, but that's fine. As long as I'm within a few degrees of 65 degrees Celsius, everything will still work, and it'll work pretty well. Okay, so there you go. Now I've truncated these primers. That's everything I need to have there. And notice over here, I've got the estimated melting temperature for each of them, but the software also estimates that the best annealing temperature for this reaction is actually a little bit above that melting temperature at 67 degrees Celsius. So you want to take note of that whenever you're designing your primers, what that temperature is, because that's what you want to use for your PCR reaction, is this temperature right here. Sometimes that might be a little bit higher than it should be, so you might want to try 66 or 65 if that temperature doesn't work for you the first time through, but this is usually a pretty good estimate right here. Okay, so good. Now I've got that. I've truncated my primers. They end in a C or a G, and it's not too many C's or G's either. So the next thing I want to do is I want to check to see if these primers do anything naughty. So what I mean by that would be I want to make sure that this primer, for example, only binds to my target sequence, nowhere else in the genome. If it binds somewhere else, then it could start copying other sequences, and I really don't want that. So what I can do here is I can take that sequence and load it into this tool, NCBI BLAST. Now what BLAST does for you is it allows you to take a sequence. So what were we talking about here? The reverse primer. There we go. I can copy that reverse primer sequence and paste it into this field. And then I can scroll down a little bit and I can say I want to look for that sequence in the human genome. Okay. Now if you were trying to check for off-target binding in a plasmid, you could click this box right here, and then you could search for this sequence in any other sequence that you wanted. So you could put a plasmid in here if that's what you're using for template DNA, for example. But in this case, we're going to be using chromosomal DNA as a template. So I'm just going to click this box right here, and it's going to search the entire human genome for any off targets for this primer. All right, so let's hit blast. This might take a few seconds, but here we go. This is what the output of that looks like. All right, so if I scroll down here, it tells me all the different places this binds, and what I want to see, first of all, is that it does bind to my target. So remember, we're amplifying the promoter for interferon lambda 1, and there we go. I've got 100% coverage, 100% identity to that promoter right there. All right, and there's a few different ways I can look at this. So there's my IFN lambda. It's showing up as an mRNA result and as the genomic DNA result, so it's good to see both of those. All right. I can also look at the alignments. So this is how it actually looks like when it's aligned to the genome. Well, in this first case, it's the mRNA. So it aligns to the mRNA and aligns to the genome. All right. Oh, sorry, I'm highlighting the wrong thing there. So what I was trying to say is that it does bind perfectly to my target sequence. That's this interferon lambda hit that we see up here. But there's always going to be other places where there's some complementarity to your primer. So for example, this other locus down here that is for a looks like non-coding RNA, you can see our primer, that's the query here, does have a lot of complementarity to this sequence here. But what's important to point out is that, first of all, it's not perfect complementarity. There's a little bit of a gap here. Actually, I'm not sure why that's showing as a gap. That should be C binding to G. That should be fine. Well, anyways, even without that gap, our primer is supposed to be 22 bases long. All right, so up here we can see that the entire primer binds to my target from 1 to 22. But down here, we only have binding from 1 to 19. So that's relieving, because if the last 3 bases of my primer do not bind to this target, DNA polymerase will do nothing with it. The last bases on the 3' end have to be absolutely complementary. Otherwise, nothing will happen. Your primer might get, let's say, distracted by this other target site that it could bind to, but nothing will come of that. It might bind to it transiently, come back off, and then go bind to its actual intended target. So this is a result here that might be scary, but it's imperfect enough that I'm not going to worry about it. If we had the same kind of base coverage here, 19 bases, but the last base, so let's say this was 3 to 22, if that were true, then I would be worried that this primer might bind to that other sequence and be extended from that. And I'd probably redesign the primer. But there we go. I don't see that situation, so that's fine. Now down here, we do see a situation in which the last few bases of our primer bind. So we have base number 22 here binding perfectly, but it's only 13 out of my 22 bases. So what that means is I've got a floppy end over here, and this interaction is not going to be as strong between the primer and this site as it is between the primer and the intended target. So as long as I keep that annealing temperature high around that 67 degrees Celsius that the NEBTM calculator had, this probably won't happen at all. This is something that would happen at lower temperatures. That primer might bind to this site, but it won't happen at around 67 degrees Celsius. Okay, so we've got 2 very good examples of potential problems, but this one isn't a problem at all because I don't have binding, perfect binding on the 3' end, and this one isn't a problem because I don't have a large amount of bases that are binding here. Okay, so then that looks pretty good. I can come back into our PowerPoint and I can say there's no major risks of off-target binding for this reverse primer here. So once I've cleared that step, I'm also going to check for any potential secondary structures in that primer. So it's automatically logging me in here. You do have to create an account for this website, but it's a pretty simple process. It's free. There are other web tools that you can use. I just like this one the most. So anyways, I can come in here and I can copy the sequence of my primer. Alright, I'm just going to copy that real quick into this field, and then I can look for these types of secondary structures over here. So for example, I could look for any hairpins that this primer might form. I click on that hairpin button, I scroll down, and it tells me, well, yes, technically you could get this very funny looking structure. Really it's just 2 C's and G's that are binding to one another. But we really don't have to take that seriously because it doesn't even form above a temperature of 36 degrees Celsius. That's what this column represents right here. That's the melting temperature of that hairpin. We're going to be doing PCR, which is going to be at a temperature of 98, then 67, then 72 degrees Celsius, so this primer will not form. But if we saw a hairpin here that had a melting temperature that was much higher, something around, let's say, 50 or 60 degrees Celsius, that might be a cause for concern, and it might be a good enough reason to redesign that primer completely. But in this case, it's really not a problem. And anything below 50 to 60 degrees Celsius, I disregard. Okay, so hairpin doesn't seem to be an issue for this. Let's check self-dimer now. This would be the chance that that primer can bind to itself in solution. And so this is what the program comes up with, is a bunch of these different structures where potentially you could have some binding between 2 of the same primer. So G's bind to C's, things like that. All right, but the way you want to decipher this is, first of all, you want to look at this sequence up here. This is your primer sequence, and if it binds to the target, what this is telling you is that the delta G of that process, this is the Gibbs free energy, okay, so that's a thermodynamic value that tells you how spontaneous the process is, essentially. It's telling us the maximum delta G is negative 45.65, okay? So that is pretty much the strongest interaction that you can have here. If I look at one of these self-dimers, the delta G values for them are much lower. Because there's only 3 base pairs participating in this reaction, the delta G is only negative 5. So since this value, negative 5, is so much lower than negative 45, I'm pretty certain this is not going to be problematic, all right? This might form transiently, but it'll fall apart very quickly, especially at those elevated temperatures in PCR. When I would start to get concerned would be if this value is anything more than 25% of this value. So if this were something like, let's say, 15 kcal per mole here, negative 15 kcal per mole, then that might be grounds to redesign my primer, because what could happen in that solution would be that the primers, instead of binding the template, would bind to themselves, and that would inhibit the entire PCR reaction. Okay, but in this case, everything looks pretty good. So we can check that box off for our reverse primer. And I do like to note what the TM of the strongest hairpin is, and the delta G for that strongest self-dimer. All right, just for future reference in case I want to look back at that. Okay, so if we want to now do the same thing for the forward primer, I go back into Blast. There we go. I'm going to copy that forward primer sequence. Go back up here to Blast. There we go. Paste in that forward primer sequence. Again, we're searching the human genome. Hit Blast here. All right, this is going to take a few seconds. But there we go. We've got some results here. And uh-oh. The first thing you should notice is that the top result here has nothing to do with interferon lambda. It's this other protein ZNF79. And there's a query coverage and percent identity of 100% to that other target. Oh boy. And if we go over to the graphic summary here, we see a lot of green bars. So what this is telling us already is that there's a lot of other places in the genome where this primer sequence could bind. This ZNF protein, for example, looks like this gamma glutamyl hydrolase, and a lot of other examples. And that primer is 26 bases long. We've got 100% complementarity over the entire stretch of that primer to these different proteins, so these other spots in the genome. So what BLAST is trying to tell us here is that this would be a very bad primer to use. It would probably, if I scroll down here, I could find it binding to our intended target sequence as well. But if it's binding to all of these other sequences, you're simply going to be spreading that primer too thin. And not only are you going to be distracting that primer, you're also going to be generating or copying other sequences as well. So this reaction probably would not work at all for this reason. So what I'll have to do then is I'll have to come back in here to my sequence. Oh boy. And since that forward primer had so many off-target binding sites, I'd have to move it again. So in this case, I could do either one. I could move this primer upstream or downstream. In this case, I'm going to move it downstream because honestly, 1500 bases is probably overkill when I'm trying to study a promoter. Most are closer to, let's say, 1000 bases. So I would choose to move this right, to the right here, downstream. Now, I've already done this. I did this example before this, recording this lesson, and what I found was that there's actually a large sequence here that is complementary to other parts of the genome. So that was painful. I had to check a lot of sequences. I had to do a lot of blasting over and over and over again. So, for that reason, I'm now going to show you a different way to do this process. So instead of doing this manually, you can also use a web tool for it. There's several apps online that will allow you to put in a sequence like this promoter here, and then it will design primers for you based on pretty much all the steps that we've already discussed. So I definitely wanted to see that for you to see how all this works, how all these primers are designed. But now I'm going to show you how to use this tool right here, Primer 3. This is pretty much what everyone uses to design PCR primers. Now I'm going to show you how that works. Okay, so there's a lot of information on this slide, but let's just jump right over into the web page itself. Okay, so this is what the tool looks like. From top to bottom, what you want to do is you want to tell it what organism you're working with, Homo sapiens. So just like we were blasting those primers, it's also going to blast the genome for primers that it designs. So it needs to know which organism you're talking about. Okay, and then we're going to take our putative promoter sequence. We're going to copy that. Here we go. All right, we got that copied, and we're going to paste that into this field. So that's going to give Primer 3 an idea of what kind of sequence I want to copy. And then if I scroll down farther here, there's a lot of information that you can put into this program. A lot of different variables that you can set as to where you want the primers to bind, how large you want the PCR product to be, etc. You can force the primers to start in specific regions here, for example. But all we're going to do in this exercise is we're going to input just a few parameters. So first of all, I like to change the primer size a little bit. So minimum 18, maximum of about 30. I like to allow for a larger primer because I prefer to have a higher primer TM. So you can see here the default's around 60 degrees Celsius, but I like to be between 60 and 70 with an optimum around 65. And to get there, I might need that longer primer size there. That's why I increased that. Okay, and then the next thing I'm going to do is I'm going to change the product size range. Okay, because what Primer 3 is going to do by default is it's going to take that sequence and try to amplify pretty much the shortest sequence it can, something that's around 100 to 200 bases. But the purpose of our experiment is we want to study that entire sequence as a promoter. So I'm going to put in here something like 1400 to 1500 bases. I want to get most of that sequence in my PCR product. So I'm forcing the program to look for primers that copy most of it. Okay, and again, there's a lot more information that you could put in here, but we're just going to click this button right here, pick primers. Okay, so there we go. It looks like Primer 3 is having the same problem that we were. It can actually find primers for this sequence. Now, it might have very well found a good primer or a good reverse primer like we did, but then it's struggling here with the forward primer, struggling to find a sequence here that's unique to the rest of the genome. So let's go back and change the PCR product size. Let's give it a little bit more wiggle room. Let's say 1300 to 1500 bases. Whenever you're doing this for your target sequence, you should definitely start asking for a larger sequence and then just go down like I just did there. Okay, so good. Now that we've relaxed that requirement here, it says it can amplify 1300 bases of that sequence, and it can do that by using a primer here. So you can see it had to go 130 bases into that sequence before it found something that was unique, and that's the sequence right there. It's also up here. You can see the estimated TM is 63 degrees Celsius, but again, it's using its own parameters to estimate that TM, so you want to actually disregard that, and you would have to go and copy this sequence and put it into NEB software to get a real TM. So this is what would actually be happening in our reaction. So instead of that 63, now it's 66 degrees Celsius that we have there. Same thing for the other primer. It estimates a temperature of 62 degrees, but if I go to NEB's website, it's actually 64. But still, I would select an annealing temperature for my PCR reaction of 67 degrees Celsius. Okay, so disregard these TMs, but anyways, there we go. We've got our primers, and we didn't have to do very much work there at all. We just gave the program the sequence, and it gave us the rest. Alright, so let's just scroll down here, see what other information we have. There's the forward primer binding site. Okay, so we are chopping off a little bit of that, the beginning of the sequence, but then it looks like we're getting, oh good, pretty much almost all of it. So almost all of the sequence down to base 1500. We are losing a little bit, but Primer 3 has deemed that necessary. It said that this is the best primer that I can make for you on this side of that product. Okay, so there we go. Now we've got those primer sequences. We can come back into the PowerPoint here. Alright, so that's all the steps that we just did. And there's the products. That's just screenshots of that. Just as a reminder, disregard these TMs. You want to go to NEB's website to get some actual TMs. And for our PCR reaction with these primers, the annealing temperature is going to be 67 degrees Celsius. Okay, so now we've got the complementary region of those primers. We still have to add restriction sites to those primers. Now, the way you want to go about this is the first thing you have to do is you've got to look at the plasmid sequence. So if I'm copying this sequence into this plasmid, I want to replace the EF1 alpha promoter that's shown here in blue. So right now in this plasmid, the EF1 alpha promoter is driving expression of GFP in green, and then here's the poly A, or the terminator sequence for that. Okay, so that's what you're looking at. But I want to cut out this EF1 alpha promoter. So I need to find a restriction site that's somewhere in this range up here, before the promoter, and somewhere in this range over here, downstream of the promoter. Okay, so a very easy way to do that is to go to this website right here. Again, we're going to go to New England Biolabs and use one of their tools. They have a lot of fantastic tools. But again, there's other websites out there. I want to be fair. Okay, but anyways, I'm going to take the sequence of that plasmid. Here we go. So I'm just going to copy all of that, Control C, and then Control V to paste it in here. I need to tell the program that this is a plasmid, so it's circular. That's just going to append the end of the sequence to the beginning. And then I'm going to submit. Alright, so what this has done is it's examined our plasmid in its circular form, and it's looking for restriction sites. Okay? Now, here we have to remember that the promoter was, let's see, what was it? The promoter is here at the beginning. So we're looking for a restriction site somewhere here in the first 100 bases, and then again around base 1200. Okay, so this works clockwise. So base 1 is right here at 12 o'clock noon, and then as we go clockwise, the numbers increase. Now, unfortunately, what I'm looking at here is that it looks like we don't have too many options upstream of the promoter. Alright, so this SSP1 is actually inside the ampicillin resistance gene that's in the backbone. And notice there that it says blunt. That means it's a blunt cutting restriction enzyme. And you can watch the restriction enzyme video for more information on this, but we generally want to avoid blunt cutting enzymes. They make life harder than it needs to be. They don't make sticky ends, essentially. Then we've also got this SEX-A1 here. This enzyme looks like it might be a good candidate, but if we take a closer look, it recognizes this CCWGG motif right here, and that can actually be methylated in bacteria. So I believe it's this C right here that gets methylated. And if that's methylated, then this restriction enzyme does not cut that sequence. So since we'll be working with plasmids that come from bacteria, we can't really use this restriction enzyme. It would not work. All right, so that's not an option. Our first option appears to be this MFE1, CAATTG. However, look at where it is. It's at base 167, which is actually, if I come into here in my sequence, that's actually inside the EF1 alpha promoter here. So I could use this, but I'd be leaving a little bit of the EF1 alpha sequence in the plasmid, and that might contain transcription factors that would contribute to the transcription. So I wouldn't be really looking at the effects of my promoter, I'd be looking at the effects of a fragment of the EF1 alpha. All right, so anyways, MFE1 is there. It's an option that we can use as a last resort. But let's say we don't want to. Well, we can keep looking. And instead of looking at enzymes that cut the plasmid once, we can take a chance and look at enzymes that cut the plasmid twice. So that would be these two cutters down here. Right, now typically you don't want to use a two-cutting enzyme. I'm only doing this because we don't have any other options. But what I'm hoping for here would be that I have an enzyme that cuts twice, once before the promoter, and then again either inside that existing promoter or after it. So if I take a quick look here, I do find that indeed there is another enzyme, SAL1, that cuts very close to the beginning of this plasmid at base 62, but it also cuts down here at 1290. And if I take a look at my sequence, where those sites are would be right here. Okay, so that's great. SAL1 cuts in this region upstream of the promoter, so I can cut all of that out. Oh, but look down here, it also cuts downstream of the promoter. Okay, so that's pretty interesting. If this second SAL site was anywhere else, if it was in the GFP gene and the poly-A sequence, then I couldn't use this enzyme because it would be taking out more of the plasmid sequence. But since it cuts where I want to cut anyways, here and here, before and after the promoter, this might actually be a pretty good choice for me. I could just use SAL1 to cut in both places. However, that's also a bad idea. If you use just one restriction enzyme, there's two bad things that can happen. First of all, after I cut out the promoter here, the plasmid can actually ligate to itself. I'll talk more about that in our restriction in my video on restriction enzymes, but what you need to know now is that if I cut with SAL1 here and here, I can end up with a new plasmid that seals in on itself and just doesn't have any promoter at all. Doesn't have the old or the new promoter. So that's kind of a bad idea. I don't want to use just SAL1. But for now, this worst case scenario, it looks like I might still have to use SAL1 to cut, at least upstream of the promoter. And then what I would hope for is that I can find additional restriction sites down here after this other SAL1 site that would allow me to prevent this plasmid from linking back to itself. Oh, I forgot to mention the second bad thing that can happen. Also, if I were to use SAL1 twice to put in the new promoter, if the new promoter did make its way in, it could go in backwards as well as forwards. There would be about a 50% chance of it going in the correct orientation or flipped. Again, I'll talk more about that in the restriction enzyme video. But another reason not to use just one restriction enzyme for cloning. Okay, so now we've got some choices. They're not perfect, but we have some choices. SAL1 and MFE1 for cutting upstream of the promoter. Now let's look back at the website and see what is available for downstream. So go back here and click the one cutters. And we're looking for around base 1200. And here we go. Looks like we do have a few options there. So at least downstream of the promoter, there's several enzymes that we could use. ECOR1. I'm going to skip over these ones with asterisks because they can be affected by methylation. But we have ECOR1, KPN1, or, well, I guess EXMO1's not too bad. So XMA1 is blocked by a different type of methylation, CpG methylation, and that doesn't occur in bacteria. So we could actually still use XMA1. So we got 3 options there. So let's map those to the sequence. We come back here, and this is where those are. So, very importantly, the first thing I need to note is that SAL1, if I use that, I can't use ECOR1, because SAL1 would cut here and here. That would remove the ECOR1 site. That would be lost. So then I'm limited to only using KPN1 or XMA1 if I use SAL1. So I could use SAL1 here and KPN1 there. The plasmid could not ligate to itself because the sticky ends here and here would be different. So that's actually looks like a pretty good strategy. I can use SAL and KPN, or I could use SAL and XMA. Or if, let's say, SAL is not a choice, I could have also used MFE1 and ECOR1. Or wait a second, actually, if we look at MFE and ECOR, they have the same 4 internal bases, AATT, AATT. And so what that means is that these enzymes might create the same sticky ends, and I would have the same problem that I had before when I was using SAL1 to cut twice. So actually, I would not want to use MFE1 with ECOR1, but I could use MFE1 with KPN or XMA1. So there we go. We've got some options. That's good enough to get started here. Okay. So there is what the sequence looks like, and just to reiterate, we can use SAL1 with KPN or XMA, or MFE with KPN or XMA. We cannot use ECOR1 because it's what we call an isoschismer. It creates the same sticky ends as MFE1. Okay, so now that we've got 2 potential strategies, now we have to look at the sequence that we're trying to amplify. So, based on the primer 3 results, remember we have those primers designed. They didn't amplify the entire 1500 bases of the promoter, but they did amplify was around 1300 bases. That's this sequence down here on the slide, okay? We want to check that sequence for these restriction enzyme sites as well. We don't want any of them to be there, because remember, what we're going to do is we're going to make a PCR product, and then we're going to cut that PCR product with these restriction enzymes on the ends. If those restriction enzymes cut inside the PCR product, then we'll get garbage. We'll get these tiny little fragments that don't clone into the plasmid. Okay? So what we want to do is we want to check that promoter sequence, the target sequence, for these restriction enzyme sites. And sure enough, we find that this putative promoter has an MFE1 site and an XMA1 site. So we could not use either of those enzymes for this target sequence. So that leaves us with SAL1. We use SAL1 and KPN1 for our cloning. Okay? So since we know that, then it's just a simple operation of taking the sequences that we have from primer 3, and we're going to take the forward promoter that's at the beginning of that, or sorry, we're going to take the forward primer that's at the beginning of that promoter, and we're going to add on to that the SAL1 site, GTC, GAC, all right? Because remember, the way this plasmid works is there's a SAL1 site upstream of the promoter, and there's a KPN1 site downstream of the promoter. Okay, so then the SAL1 site is going to be on the forward promoter, and there's my protecting bases. And notice there's no complementarity between them and these first 4 bases. All right, and then for the reverse primer, we've got our other primer 3 results, and to that we're going to append the KPN1 site, GGTACC, and another 4 protecting bases. So there you go. That's all you need to do there. And once you have these sequences, you can submit those online and order these sequences from a company, and 2 days later you get plenty of primer to do your PCR reaction. So that's exactly what I'd like you to do, is look at your putative promoter sequences, design some of these primers, and submit those to me, and I'll order them for you. All right, but if you run into any problems, so let's say all of those restriction sites that are listed there in step 4 bind to your promoter, let me know, and we'll figure out a way to solve that problem. All right, good luck, and let me know if you need any assistance.